This lesson is all about mitosis. Mitosis is what is frequently referred to as cell division. Mitosis occurs in every cell, every living eukaryotic cell. Mitosis is the process by which one new cell produces an identical cell just like it. As you can see from the definition, mitosis is when a cell divides and produces two identical cells, just like it. Here are a couple of key terms that we will use throughout this lesson and in several of the other lessons that we will have down the line here, as well as even some future units. First is the term chromosome. Scientists define a chromosome as basically a long strand of DNA wrapped inside of a protective protein coat. Different organisms have different numbers of chromosomes. For example, humans have 46 chromosomes in their cells. The next term is chromatid. Now, chromatids are important to understand because they only occur as a cell completes the replication process and begins to carry out mitosis. Chromatids are defined as two identical DNA sequences produced through this replication process, held together by a piece of protein called a centromere. As you can see in this drawing, the centromere kind of acts like a rubber band, a twisty tie to hold the two identical pieces of DNA in one location, hold them together. What this does is this permits the cell to move these identical pieces of DNA in unison during the early stages of mitosis in order to ensure that mitosis occurs correctly. The final term is interphase. Now, scientists use this term interphase to describe what the cell is doing when it's not carrying out mitosis. One thing to understand is if you think about a cell carrying out mitosis for approximately every 24 hours, the mitosis process only takes roughly 1 hour or 80 minutes of that process. Well, the other 23 hours is interphase. So a muscle cell is contracting and relaxing to move the bones during interphase. A pancreas cell is producing insulin during interphase. Okay. So cells are doing what they're designed to do during this stage of interphase, and at the end of the interphase process, it's when the cells will actually copy their DNA in preparation for the process of mitosis. Now we can begin to talk about the steps of cell division, or the steps of mitosis. The first step in the mitosis process is known as prophase. Scientists use this term this prefix pro to describe as the fact it's the first stage of mitosis. At this point in the process, the cell will basically take apart the nuclear membrane in preparation for moving the chromosomes around. And it's at this point that the chromosomes, the DNA, will coil up into these chromosomes as they are getting ready to be moved around, so they become coiled up like springs. What this process does is allow the DNA to be moved without any harm to the DNA. At this point in the process, the centrioles become active. These are an organelle that basically are in a resting stage until mitosis kicks in. Centrioles will produce a series of spindle fibers which will grow in length and eventually attach themselves to the various chromosomes in preparation for moving the chromosomes about within the cell. And you'll notice this final picture shows a onion cell And it shows an onion cell in the prophase stage. So you can see the nuclear membrane is gone, the chromosomes are coiled up. If you look at the cell above it, the cell above it shows a cell in interphase, and you can see the nucleus and the nucleolus in that cell in order to be able to differentiate the two of them. Once the cell has completed prophase, the cell will begin to enter now metaphase. Meta, prefix means middle. And so in metaphase, what will happen is the spindle fibers will attach to these chromatids, these pairs of chromosomes, and manipulate them in order to line them up along the equator or along the middle of the cell. Now this is again in preparation for the continual eventual movement of these chromosomes in order to create two identical groups of them at the end of mitosis. Now in this picture here you can see again this onion cell, and you can see the chromosomes basically aligned with that central equator of the cell. And so if you look close at the picture, you'll also be able to kind of see the spindle fibers attached to those chromosomes. And again, once this occurs, the cell will have then completed the metaphase portion of mitosis. Upon the completion of the metaphase stage, the cell will now enter anaphase. Again, ana, away, prefix meaning to separate or to move away. And again, in this anaphase stage, it's at this point that the cell will use enzymes to break down the centromeres, and this will free up the individual chromosomes. Remember, the centromeres hold together two identical chromosome strands, and so now each of those strands are free to move, again being pulled by those spindle fibers to opposite poles of the cell. Now this picture here shows this occurring, and so you can see in this picture of the onion root, okay, that you now have two groups of chromosomes, and if you look at them and kind of visualize this, you can almost see them being dragged through the liquid cytoplasm of the cell as they move towards each end of the cell in this stage. And now, finally, the cell is ready to go through telophase, the final stage or step of mitosis. It's in this stage that those chromosomes, those visible dark purple chromosomes we've seen in the other slides of the onion cells, begin to uncoil and return to the chromatin state in order to permit the cell to read the DNA. And that's important because it's as the cell begins to close out the telophase stage, the cell will carry out a process known as cytokinesis. And in the process of cytokinesis, the cell will reform the nuclei, and the cell will begin to make the various organelles that are needed for the cell to function correctly. Now in this picture here, you can see a cell beginning to close out that telophase stage. You can see running down the middle of that cell between the two forming nuclei the beginnings of a cell wall. At each end of that growing cell wall, you can see the remnants of the spindle fibers that were used to move those chromosomes to where they needed to be in order to create, again, these daughter cells. Now, once a cell has completed this process of cytokinesis, it will, again, create the two nuclei that are in each of those two cells, and as a result of this creation of the nuclei, two identical, though likely smaller, daughter cells will have been created. Now, this will vary somewhat depending on the type of eukaryotic cell we're talking about. If it's an animal cell, what will happen is, okay, various parts of the cytoskeleton will pinch together, pull on the cell membrane to pull the membrane together and cleave. The word is cleave, or cut the cell into two. In a plant cell, such as in this drawing that's now here, you will see that the cell just creates the new cell wall, and once that cell wall has been built to separate the two nuclei, the two daughter cells now are present, and again, mitosis will have been completed. Now again, to just review for a moment this whole process of mitosis. One, it's important to understand that the process of mitosis takes about 80 minutes to complete. As you can see in this slide, not all of the stages are equally long, okay? Metaphase, okay, is much longer than anaphase, for example. You know, interphase is, again, not as long as metaphase. So each of these stages in total comes to about 80 minutes. And on average, a cell may divide approximately every 24 to 48 hours. So as you can see, this process of mitosis is only a small part of the cell's life cycle. And that's important, again, to understand. Before we move forward, I would like just to, again, review these with you, because it's important, I think, for you to understand that these 4 stages or 4 steps of mitosis are universal in nature, meaning plant cells, bacterial cells, animal cells, protist cells, all carry out this same process. And again, it starts with a parent cell undergoing prophase, destroying that nucleus, basically wrapping up the chromatin into chromosomes so that they can be easily manipulated and moved within the cell, and then entering metaphase, upon which the chromatids, those pairs of chromosomes, will then move to the main equator of the cell with the help of the spindle fibers created by the centrioles. Once that's occurred, then anaphase begins, and anaphase basically has the spindle fibers pull those chromosomes to opposite poles of the cell, and then entering, lastly, telophase, whereby the cell will eventually divide into 2 identical daughter cells upon the completion of the cytokinesis process. As I begin to close out this lesson, I want to talk about 2 other things with regards to mitosis. First, what is the function of mitosis? Well, it depends on the type of organism that you are. If you're a single-celled organism, such as a protist or a bacterial cell, basically this is your method of reproduction. You just clone your cells. You produce identical copies of your cells in a quick and easy manner, again, permitting you to produce lots and lots of organisms of your species. Right? And so, again, single-celled organisms, it's a means of asexual reproduction. Now, if you're a multicellular organism, such as animals, such as yourselves, or plants, fungi, mitosis serves a different purpose. Mitosis will serve the purpose of basically organism growth. It will increase the total number of cells within the organism. I mean, that's what's been going on inside you, along with differentiation of those cells into unique types of cells since you were born, or since you were conceived, I should say. Excuse me. And so it increases the number of cells, allows for organism growth. The second purpose of mitosis in multicellular organisms is to replace old or injured cells. When cells reach the end of their life cycle, or when cells are destroyed, they are usually replaced, or they are replaced through the process of mitosis. I mean, the easiest evidence that you have of this process is through getting a cut on your finger. You have a cut on your finger, you put once the bleeding stops and a scab forms, what goes on underneath that scab is mitosis. And once the cells have been rebuilt and the skin has been reformed and recreated, the scab form falls off, and your skin is back as good as new. And so, two purposes if you are a multicellular organism: growth and repair and replacement of injured, destroyed cells. The last thing I would like to talk about is how does mitosis differ between plant and animal cells, excuse me. First of all, plant cells do not have centrioles, okay? And so this organelle is unique to the animal eukaryotic cell. Second is that cytokinesis takes place slightly differently in plants due to the cell wall process. And so, generally speaking, cytokinesis occurs, okay, once the cell wall has been created in plant cells. Cytokinesis usually occurs once the two cells have cleaved in animal cells. And so there's a slight difference between when cytokinesis occurs in plant and in animal cells. And that wraps up this lesson in mitosis. Again, just to review very quickly, mitosis, okay? Process of producing identical daughter cells, 2 identical daughter cells, 4 steps: prophase, metaphase, anaphase, telophase. Okay, prophase, basically release of the chromosomes to allow manipulation within the cell. Metaphase, movement of those chromatids within the cell to the central equator of the cell. Anaphase, destruction of the chromatids to allow movement of the individual chromosomes, equal numbers to each pole of the cell. And then finally, telophase, with the process of cytokinesis at the end of that stage, producing 2 identical daughter cells with the same number of chromosomes as the original parent cell.